Welcome to Clinical Trialblazers, a podcast by Parexel Biotech, where we dive deep into the journeys of industry leaders shaping clinical research and biotech innovation. I am your host, Alberto Grignolo. In this episode, I'm joined by Miquel Vila-Perello, co-founder and CEO of SpliceBio, a gene therapy company based in Barcelona, that he has led since its inception in 2014. Under his leadership, SpliceBio has grown from an early-stage concept into a global clinical-stage biotechnology company. The company has advanced multiple programs into development, including the lead program for Stargardt disease that is currently in the clinic, and secured over $200 million in funding from top-tier investors. Today, we will discuss Miquel's career journey from academia to the biotech startup world, as well as the challenges and opportunities in developing gene therapies, the lessons he has learned in obtaining funding, and his insights on the future of biotech innovation. Miquel, it's a pleasure to be with you today. Welcome.
Great. Thank you. Thank you for the nice introduction, Alberto. Also, a pleasure to be here with you in the podcast.
I believe you are in Barcelona. You live and work in Barcelona, right?
Yes, indeed. Born and raised.
My favorite city. Love Barcelona. So let me ask you about Catalonia. Rightly or wrongly, I have the impression that Catalans have a reputation for being fiercely independent in their thinking, maybe independent in their actions sometimes. So do you consider yourself to be an independent, perhaps unconventional thinker? And if so, how so?
Well, I think it's a kind of an interesting way of seeing us. I think actually one of the key characteristics of Catalans, or the way we define Catalans ourselves kind of culturally, it's by having this mix of what we call seny and rauxa. This is a difficult thing to translate, but seny, more or less, it's equivalent to common sense, reason, and rauxa, it's the opposite, kind of passion, impulse, almost to the level of frenzy. And, culturally, we're supposed to have a mix of both things. And I think, you can see some of the rauxa in many of the famous Catalan artists, such as Salvador Dalí or Joan Miró. In myself, I think I can see a little bit of a mix of both. So I typically would be tempered and conscious when making decisions. I would lean towards the conservative side of things, but I've also shown a little bit of signs of this impulse and passion. I think a clear example is when I decided to start SpliceBio, my decision to quit, a pretty stable and good job at Princeton University, right? Obviously a top worldwide university, and move to Barcelona at that time with basically just enough money to pay a year of my salary and that of one of the co-founders. So I'd say that was a pretty daring and passionate, impulsive moment. But then through the journey of SpliceBio, we've had to combine, again, these two aspects of the Catalan identity. We need to be passionate and show impulse and we need to stay true to our goals. It's a challenging journey, so you need a lot of that. But at the same time, you need common sense. You need to have reason to basically guide the decisions and the different steps that we need to move forward.
Is there a tension between these two characteristics, if you will, rational and passionate inclinations? Yeah. And how do you balance the two? Have you ever sort of gone off balance? Too much rationality, not enough passion, or too much passion at the expense of logic?
I guess that's one of the dangers of this kind of stereotype of the Catalan identity. And again, I think always you'll have one more dominating than the others. In my case, I'd say kind of the reason side of things tends to dominate. But yeah, there's a risk there of, this over-analysis. We call it, paralysis by analysis, right? If you try to overthink things too much, that could be a negative thing. And often in the biotech industry, and I'm sure that in other industries, it's the same. At some point, you need to make decisions with information at hand. And although, by personality, I would like to have, all the information possible, the reality is that at a given point, you have the information that you have, and you need to make a decision, and you need to feel comfortable with that situation, right? Sometimes having to make decisions without having all the information that you would like to have.
As a native Italian, I know about passion and being sentimental. And I think in biotech, it's important to be passionate about the mission and to actually pursue it through the ups and downs and inevitable challenges and difficulties. The passion can actually carry one pretty far, wouldn't you say?
Yeah, no, I agree. There's very tough moments in drug development and in the biotech industry. And if you don't have the passion to stand by what you believe or what would you think it's the right thing to do, then those difficult moments, it would be difficult to keep going. So you do need a lot of that because I think again it's a challenging industry, and the way we can take these complex products forward through development requires someone believing in what they're doing and really at some points just pushing through.
Well, let's cheers to passionate Catalans and best wishes for success. Let me move to your academic experience. You've been both a student and a researcher prior to crossing into biotech, and you accumulated that experience on both sides of the Atlantic Ocean at Barcelona University, Rockefeller, Princeton. So I want to ask you about what insights you might have picked up about differences and similarities in the approaches to research in these two different cultures.
I was actually lucky to work on top tier research groups on both sides of the Atlantic, both in Barcelona and then Rockefeller and Princeton. And actually, my PhD advisor in Barcelona was trained for many years in the US. And I think he brought in already in our Barcelona lab some of the ways of doing things in the US. So to be honest, I didn't notice any or didn't experience any dramatic differences. It is true that back then, and maybe the situation has changed now, the amount of resources that we had in our labs in Barcelona was quite different than what you had in the US, right? So in Barcelona, we often had to find, workarounds to accommodate for the fact that maybe we couldn't buy a specific reagent because it was too expensive, right? And then we would need to find a way to either synthesize it or prepare it ourselves, or a piece of equipment that, again, it could be expensive, and then we would need to find a way to kind of make some homemade stuff that we could use in the lab. While in the US, when I moved there, that was 2005, the amount of resources which was much bigger than what we had in Barcelona, and there you could really focus on trying to address the question in hand. What I learned in kind of when I was working with more stringent resources was also valuable, right? Because related to what we were saying earlier, it allowed me to, when you hit a wall or you find kind of this problem because you don't have enough money, then you know you can find ways around it. Obviously, it's going to take more time. It may take some extra effort. It may take more time to get you to the answer that you're actually trying to answer with the experiment, but it's doable. So I think being exposed to the two environments, I think I see it as a positive thing in my own training. And I think for all scientists, it would be good to be exposed to both things, right? Kind of the fast-paced, really science-focused that we could do in top-tier research institutions in the US, but also at some point being exposed to, well, how are we going to address this problem when you don't have a big amount of money that you can buy, any reagent that you would want. You'll need to find other solutions, right? Not just checking the catalog and buy that thing that you want to buy.
I want to ask you about your experience in moving from academia to industry. That kind of transition can be quite challenging for some people. In my case, when I moved from the lab to the pharmaceutical industry, I realized, okay, suddenly I have to worry about budgets, timelines, deadlines, regulations, a lot of other things. In your case, how was your experience in moving from being an academic scientist to eventually becoming a co-founder and biotech CEO? Are these two worlds so different that you had to fundamentally change your mindset? How was that?
Yeah, I don't think I had to change my mindset. I think I could find some common characteristics of both worlds. I mean, you're totally right that some things like timelines, budgets are critical in companies, while in academia, unless you're a PI that are you also managing those budgets, you're less exposed to that, right? I think that's definitely a difference. In other aspects, I think, and going back to some of the things we were saying earlier, I think in both worlds, resilience, keeping at it, and remaining true to your goals are common characteristics of both being a CEO of a biotech or being an academic researcher. Both in basic research and in drug development, projects take a long time. It may take a long time to prove your hypothesis or take your drug to the next stage of development. And you will be faced with a lot of setbacks and difficulties. So you need to have the mind able to go through those setbacks. And as we said earlier, keep at it if you think that's the right thing to do. Obviously, at some point, if you've hitting the wall multiple times, it's also smart to say, okay, I've given it my best try. I need to let go. But I think there's some common characteristics between academia and being a CEO of a biotech company. Maybe one of the differences that I struggled at the beginning is that my training as an academic scientist was to be able to address all the problems by myself. Each scientist in the lab, it's almost kind of a little company, right? You need to take care of everything related to your project. And sometimes as a CEO at the beginning in particular, it could be tempting to see your company the same way. Like you need to control, you need to know everything. And that's obviously not possible. So it's very important not to make that mistake and early on realize that you need actually smarter people than yourself and that are experts on different areas of the company, bring them on board and let them do their job because they're going to be able to do that job much better than yourself, right? But I think that can be sometimes a tendency of academic CEOs of having this kind of control mentality or this kind of responsibility feeling that you need to really do everything by yourself.
Very well said. Let's talk a little bit about SpliceBio and what you're doing there. You're focusing your initial efforts on a cure for Stargardt's disease, an inherited, rare genetic disease of the eye in which light-sensitive cells in the retina don't work correctly. So why did you decide to focus on this particular rare genetic disease at the start?
We will not get into details of the technology, but basically we have a technology that allows us to deliver large genes to treat diseases that cannot be treated by existing approaches. Okay. So when looking into which areas we could apply this technology, the eye, and in particular the retina, became a very clear, suitable target. The retina allows us to do localized administration, which means a much safer profile for the drugs because you don't need to expose the rest of the organism to the drug that we're developing. And also the fact that with this localized administration implies that we need much lower doses of the drug, which again, it has implications in terms of the costs of manufacturing. And in gene therapy, there was a good precedent. Actually, the first gene therapy approved was for a retinal disease. So there was a very good understanding on the non-clinical and clinical development of gene therapies in the eye. And as we were coming in with a new technology, it made sense to go into a tissue where some of the other aspects of drug development had been the risk. Also, it happened that in the eye, there's a lot of inherited retinal diseases that are caused by large genes. So again, that's the type of indication that our technology allows us to address. And in particular, Stargardt is the most prevalent of these inherited retinal dystrophies. So again, there's a significant unmet need, right? So everything sort of pointed to the right direction in terms of unmet need / market, as well as technical feasibility. So that's why we decided to focus in that space.
How prevalent is Stargardt, just in round numbers, more or less? How many?
So we estimate between 100,000 patients between Europe and the US. So it is a rare disease, but it's really on the really high end for rare diseases.
Yeah, right. Does your gene therapy platform actually lend itself to maybe targeting other diseases in due course?
Yeah, indeed. So the technology, it's sort of indication or tissue agnostic, and we are working in some other targets in neurology, so for the treatment of CNS disorders. So indeed, the platform could allow us to treat other indications, and we have generated a wealth of preclinical data demonstrating that indeed we can apply this to many other indications and to many other tissues.
Since this is Clinical Trialblazers, I need to ask you and want to ask you, what is your personal experience in designing and conducting clinical trials?
My background is in basic science. So this is going to be the first clinical trial that I'm involved in performing. And also related to what we were saying earlier, it became very apparent that, that's not something that you can easily do if you don't have prior experience. So we obviously needed to recruit the right people in the company to help us carry out those activities. And so we actually recruited Anis Kirak as our chief medical officer. He's actually one of the CMOs that has run the most clinical trials in ophthalmology gene therapy. We've built a clinical team so that we can take these therapies forward. So I think clinical development is indeed an area where you need strong expertise. And if you don't have it internally, you need to incorporate the right people into the team.
Let's talk a little about money. You were, of course, in academic research, which depends very heavily on grants. And so you have to apply for grants and obtain them and defend your proposals. In biotech, you have to get investors to invest in your company. SpliceBio recently secured $135 million in Series B funding, a very substantial amount. So what have you learned in this process? How would you compare that experience with trying to get grants funded at Rockefeller or Princeton? And what are your 2-3 major takeaways from your experience with biotech investors compared to grant funding agencies in academia?
One difference is not only with grant funding, but I think it's a learning also sometimes the way biotech entrepreneurs think about the fundraising process. But I think in grants it's the same, right? As you say, in grants, you write your grant and sort of you send it, it's evaluated, you may get some questions back, and either you get the funding or you don't. And sometimes the tendency could be for biotech, in the biotech industry to think that investor funding works the same way, where you kind of show a picture and either they buy it or not, so to speak. But I don't think it's at all like that. The investors typically will want to see kind of a video. They will want to see what your plans are. They will want to see how you operate in the course of a few months. They will want to see, okay, this company said they would do this, and then 3 months later, they've done what they said they would do. Obviously, if you plan to close your round in X months and you haven't allowed for the time so that the investors can see you kind of working and in progress, then they will not see this video, and then it's going to be very difficult for them to make a decision. So for me, that was an important learning. I think this is someone else told it to me. They don't want to see a picture; they want to see a video, and I think that's an important learning that I've had. Another one is how important is to build trust with these investors. The best way to do that is being transparent and really understanding that at the end of the day will end up forming a partnership with these investors. The investors, if they make the decision to invest, they will be part of the company. You'll need to work very closely with them. And so to do that, really, trust is very important. Trust from both ends. So, through the fundraising process, hopefully you can build that trust. And obviously that's something that it's not present at all in grant funding, right? I mean, you don't need to build any sort of trust with a funding agency. You just submit your grant, and sort of trust is kind of a given, right? Here, you do need to work towards building that trust. Maybe a final takeaway, maybe more on the personal level, is that obviously these processes are extremely stressful, and you need to find a way to manage that stress.
What is your way to manage stress? As a passionate Catalan, do you get more stressed or less stressed? How do you manage that?
I tend to get quite stressed because of my kind of calm and common sense mentality. I think I tend not to show it, but I do go through stress. I think time with family, exercise are things that are help me to manage stress. And obviously, when you do those things, basically keep your phone out of reach. I think you need to find some time to do that. Otherwise, the risk of burnout is very high.
In other interviews and today, you've mentioned the importance of trust, transparency, partnership with your investors. Certainly agree with that. That all sounds very nice. Do they micromanage you? Do they second guess your decisions? Do they get in your way? And if that happens, how do you manage that?
I think that's a risk. I think the second guessing part, as I was saying, that's something that is struggled earlier in my tenure as CEO. But then I also learned that, at some point, I had to make a decision. I had the information I had, and then I made the decision. And once the decision is made, I will not second guess, because that it would lead to craziness. In terms of micromanaging, I think investors, obviously, they do have access to a lot of information. They are exposed to a constant inflow of information, and they often would share some of that information with the idea and the aim of being helpful to the company. I think the line in the sand is, management is responsible for managing the company, executing on the plans, on the strategic plans that we have set and that the investors have agreed. And that's what we need to be focused in. I think it's good to receive input, because there's things that you might not have catched, or, things that you've missed, or information you didn't have. So it's good to have it, but then the investors need to realize that, okay, you have it, and then you act based on the input of your team, etc. So I don't think the expectation should be, and in our case, it's not the expectation of our investors that each time they send you an email, you need to quickly answer that email and act on what they are saying, because often they are simply sharing information they've had access to. So I think in our case, from both ends, we understand that we play slightly different roles.
Right. I would think if you have built deep trust with your investors, they might be more likely to leave you alone, saying, no, we trust Miquel, he's a man of his word, he does what he says he's going to do, keeps us informed. Is that true? Have you built enough trust with your investor syndicate that they tend to basically, okay, Miquel's got this, and we can leave him to do his work?
Yeah, I think so. It's true that, at different moments in the company, you go through new stages, or particularly, moments in which there's particularly high inflow of information or a lot of things going on. So then, obviously, one thing you need to do in terms of governance is to actually keep your board informed of things, right? You cannot pretend that, for example, when you're going in the middle of a financing, like what we recently did in a challenging financing environment, pretend the board can be kept without information for 2 months, right? They need to know what's happening. So, but I think that I wouldn't call that micromanaging. I think just it's, again, related to the trust and being transparent. And at certain moments, again, there's a lot of information, and I think it's good to keep them informed, which is, in a way, it's a way to prevent they feel the need to micromanage.
Yeah,
right?
I might call that EQ, emotional intelligence, trying to understand what their expectations are, being sensitive to them, and respond in advance to those expectations, so they won't be kept in the dark or blindsided later on, which would be even worse.
It's also one of the takeaways, let's say, of the fundraising process, right? At the end of the day, people that you interact with the fund, they also need to present the investment case to their committees, to their colleagues, etc. So you do need to understand what they need to do their job, right? So it's part of this building this partnership of, if you need to try to help them make their job, so that then they can help you do your job.
Well, that's true. I've learned in my experience that it's important to make your boss look good. So in your case, you're trying to need to make your investors look good to their bosses, management, and their own investors. Miquel, in light of recent very public controversies in the gene therapy space, what do you see as the most exciting possibilities and challenges in gene therapy going forward, including those for SpliceBio, but for the companies in this space, in gene therapy space?
Sure. So, focusing initially on SpliceBio. So for us, obviously, the ability to be able to deliver large genes, which is something that we can enable with our technology. We think this is like a key topic for gene therapy in the coming years. This is really allowing the gene therapist field to think about many targets that until now were out of the realm of possibilities. Many pharma companies, one of the first things they do when they look at a target, they look at a new disease, is, can I fit this into the viral vector we need for delivery? And if the answer was no, that target was out of the pipeline of that specific company. So I think these approaches to be able to deliver these large genes, I think it's going to be what will help gene therapy to expand. Beyond our specific area of focus, I think the area of manufacturing, it's where a lot of challenges we find, being able to produce viruses of the adequate quality and with the right yields. It's a space where there's going to be a lot of pressure from both regulators and also drug developers to be able to make these therapies a reality for patients in need and really deliver on the promise of gene therapy being this one and done treatment that could treat diseases that cannot be currently treated. But there is a need of putting a focus or emphasis in improving manufacturing processes.
Yeah. We all remember the tragic episode decades ago at the University of Pennsylvania, Jesse Gelsinger, which put a stop to gene therapy for a long time. And now it's recovered. But I want to ask you, is there a sense that gene therapy is always sort of near a cliff, that something could happen tomorrow that's going to stop the field cold for years or decades? Are we still facing such a risk, or are we past that?
I think we have learned a lot over the years. It's true that, I think in gene therapy, but we see this also in other areas of biotech. There's these molecules or modalities that at some point they become on vogue and there's this kind of gold rush to get into those areas, and then something happens and it goes down. We've seen this, for example, in the area of ADCs as well. They were
antibody drug conjugates.
Yeah, exactly. Antibody drug conjugates. They were super hot. Then they completely went out of favor, almost to the point of disappearing completely. And now they came back like really strong, right? So I think in all areas you'll find these. I think in gene therapy, there's this huge promise that, at some point with the decoding of the genome, that this could be used to treat all diseases. I don't know if that's the case. I think there's always going to be room for multiple modalities. I think gene therapy is well suited for some modalities or for some tissues, and we've been making a lot of progress. I don't see kind of a danger of a cliff that could kill the field.
Okay. Well, let's hope so for the sake of patients who are the ultimate beneficiaries of all of these wonderful efforts, including Stargardt disease and its SpliceBio. Miquel, that brings us to the end of our conversation. I really want to thank you for your insights, your candor, your openness to questions, whether serious or less serious, and really appreciate your time. Thank you.
Sure. Thank you very much, Alberto, for the invitation, and to Parexel as well for the invitation to be in the podcast.
That's it for this episode of Clinical Trialblazers. A big thank you to Miquel Vila-Perello for joining us and sharing his such valuable insights with us, particularly like your passion combined with your reason and how both are driving the progress of SpliceBio. Listeners, stay tuned for more episodes of Clinical Trialblazers, where we will continue to explore human stories behind the ideas and innovation shaping biopharma and biotech. And if you enjoyed this episode, be sure to check out our other conversations with leaders across the industry. Please take a moment to rate and review this podcast wherever you listen. Clinical Trialblazers is created by Parexel Biotech and hosted by me, Alberto Grignolo. Special thanks to our production partner, Wonder Media Network, and thank you for tuning in. Until next time, stay curious and let's keep pushing the boundaries of clinical research. Goodbye for now.